Bioremediation is the use of bacteria, fungi, and plants to break down and consume pollutants in the environment. It is a low-cost, high-efficiency, and eco-friendly method. Bioremediation is used to degrade toxic organic materials from pesticides, oil spills, industrial waste, etc., and converts them into less harmful compounds like CO2, H2O, and N2. A branch of bioremediation is microremediation, and it uses microorganisms to break down contaminants by using them as a food source, like utilizing fungi mycelium in contaminated soil sites as a remedial treatment in cases like repairing soil from wildfires, because the mycelium is both fire resistant and water retardant, and the toxins/waste are fully absorbed by the fungus typically within a few weeks. There are also enzymes such as oxidases, pestinases, and celluloses that are byproducts of fungi, and they have massive implications for cleaning up contamination. And as mycelium spreads, it secretes these enzymes that can break down pollution.
Through bioremediation, fungi present a hopeful remedy to many human-caused environmental issues, particularly when it comes to soil contamination. A healthy climate necessitates healthy soils. Plastics, which are ubiquitous in human life and in our soil, are dangerous for consumption by us and animals. Plastic pollution is going to be a major problem in the 21st century, but because plastics are hydrocarbons, fungi present a viable option for cleanup. Cigarettes are one of the most common types of litter, but they leach heavy types of metals such as lead, copper, zinc, etc. Currently, the Australian government is funding research into using oyster mushrooms to recover these metals from the soil. Other man-made catastrophes, such as oil spills or human-caused wildfires, leave behind dangerous chemicals in the soil, such as benzene, which is a carcinogen, which fungi can absorb and decompose. In the midst of a growing climate crisis, it's important that humanity, one, slows their consumption of commercial products, and two, slows the rapid change of land use change. Imagine a world in which companies use fungi as an end-of-life solution to their products, preventing them from remaining stagnant in a landfill forever. As a result, less land would need to be converted to landfills in the future, which is important considering land use change is one of the biggest causes of climate change. Additionally, removing our toxic substances from the environment leads to healthier and more robust ecosystems that can more readily withstand our changing climate.
Various fungi can be used for bioremediation. These fungi are primarily ascomycetes and basidiomycetes. A large group of basidiomycetes that are commonly used are white rot fungi. White rot fungi is an umbrella term for fungi that intentionally rot wood in order to digest it. Some common examples of this are the turkey tail and the phoenix oyster. Turkey tail are often used to lower blood sugar, boost immunity, and improve gut health. While the phoenix oyster is used to lower blood sugar as well, it can also act as an anti-inflammatory and reduce the growth of cancerous cells. On another note, an ascomycete used for bioremediation would be trichoderma. Trichoderma are typically described as filamentous fungi that are commonly used to control plant pathogens. They are also capable of growing on various different kinds of substrates.
Fungi are the backbone of natural decomposition. But how does it absorb and break down toxic materials? As previously mentioned, fungi uses enzyme reactions to break down materials. These enzymes include peroxidases, cellulosis, pectinases, oxidases, and others. These enzymes are byproducts of fungi and help to clean up contamination humans leave behind. As the mycelium grows and spreads, it secretes these enzymes, which then break down the pollution. This is an image of peroxidases. This particular enzyme is formed by the catalyst and oxidation of a substrate by hydrogen peroxide. These enzymes are great for breaking down toxic pollutants, which make mushrooms a great fit and explain why they work well for bioremediation.
One example of a basidiomycete fungi being used in remediation is the white rot fungi. Specifically, this fungi was used to treat wastewater of a food industry plant. In this case, 4 different strains of white rot fungi were used. To understand how fungi remediate wastewater, you must understand that there are 2 forms of wastewater: biodegradable, which are easily digestible simple compounds like carbohydrates, proteins, fats, etc., and non-biodegradable, which are difficult to treat humic substances. Humic is a byproduct of plant and animal tissues being broken down. Due to its complex structure, it becomes a difficult substance to treat in water plants. It is also difficult to treat with bioremediation due to its effect on the manganese peroxidase activity in fungi. Manganese peroxidase activity is a vital component in the process of breaking down lignin, a phenolic polymer used for mineral and nutrient transportation in fungi. Without lignin, the fungi will likely die in this scenario. It poses a large environmental concerns, can create harmful carcinogenic compounds like triethylamines, and are known to cause bladder cancer and can hurt industry plants financially. Humeric substances can increase the chemical oxygen demand in the wastewater. Chemical oxygen demand is a type of measurement that is used in environmental chemistry that measures how much oxygen is taken up in a chemical reaction. Chemical oxygen demand is a heavily monitored aspect of wastewater plants, and having an excessive amount can lead to rising fees for the plant. In this case, humeric levels were measured by color removal success and the size exclusion analysis. White rot fungi is one of the more researched fungi used in bioremediation due to a strong ability to degrade lignin. Lignin is typically resistant to enzyme degradation due to its unrepeating structure. Despite lignin being a source of carbon that the fungi could use for growth, they actually look for it to use the cellulose from the wood. The interesting characteristic the white rot fungi use are their extracellular enzymes, specialized in breaking down lignin. Extracellular enzymes are located outside of the cell, allow nutrients and other minerals to be broken down and then absorbed by the cell, thus saving energy. Since white rot fungi's enzymes are non-specialized, this allows for them being able to treat a wide range of pollutants as seen in the bottom right picture. Overall, this study proved evidence that the strain T. versicolor was the most successful in bioremediation due to it having 90% color degradation and a 45% degradation of humeric acid, the highest of all the strains. This can be seen in the graphs. T. versicolor is the top left chart. This has yielded promising evidence that white rot fungi is both a successful substance to use in bioremediation and, due to their abundance in nature, relatively inexpensive. Most fungi used to treat humeric substances are killed very quickly due to the damages from the humeric acids to the manganese peroxidase activity. However, the white rot fungi proved to be able to sustain these damages, going as far as continuing to reproduce and grow despite the harsh environmental conditions.
We just heard a great example of a basidiomycete proving useful in eco-friendly bioremediation of harmful chemicals and pollutants. Now I'd like to talk about another example of fungal remediation by means of an astomycete. In 2016, the University of Technology Malaysia began studying new means of remediation through fungal processes. New processes were being researched due to the large impact that industrialization has had on our environments, in particular our aquatic ecosystems. This group was studying the potential of fungi assisting in the bioremediation of alizarin red S and quinzarin green SS, two toxic industrial dyes. Next slide. The presence of these dyes in water reduces light penetration, reducing the amount of photosynthesis taking place and crippling dissolved oxygen. These dyes are also mutagenic and carcinogenic, affecting humans' health too. Recent studies using astomycetes have gained great interest due to their fast-growing nature. They also have two methods of attack, those being bioabsorption and enzymatic processes. After isolating many strains of Trichoderma elixir, it was strain F21 that was found to have the highest colonization and rapid decolonization rates. More importantly, Trichoderma elixir F21 was able to hit its highest levels of biodegradation on the 7th day when enzymes like lactase and catechol 12 dioxygenase were at peak levels. These enzymes proved they were efficient with their ability to convert ARS and QGSS into phenolic and carboxylic acid compounds without the formation of toxic aromatic amides. The results of this study proved the bioabsorption and biodegradation efficiency of Trichoderma elixir F21, making it an eco-friendly treatment that is simple and effective in treating contaminated effluent.
In conclusion, bioremediation is an important aspect to helping the environment.
And it's lots of different kinds of fungi can be used in different situations.
They use enzymes to break down certain pollutants.
Have evidence to prove their efficiency.
And fungi are a comparatively cheap bioremediation factor. Despite this, the field is relatively underfunded.